Okay, good afternoon, everyone, and welcome back to this class in microbiology. And we're going to carry on our discussion today about metabolism, and in particular microbial metabolism. But I want you to remember something, and that is that the processes that we're going to discuss evolved in bacteria, but they are preserved all the way through evolutionary time. And in our cells, we'll see much the same kinds of reactions, with sometimes with a little bit of elaboration, but the roots of all of our metabolic processes, all of our physiological processes, all lie in the bacteria. So what we learned today has application for all living things, not only for microbes. I want to just go back here. This is where we finished off last time, and I want to repeat this section because this is so important. And we've been talking about enzymes and their function, and we saw that there are ways in which enzymes can actually, the activity of enzymes can actually be affected. And one of the ways is by switching that enzyme on and off. The first way we learned about it was this. It's a system called competitive inhibition. And it's an entirely artificial situation. This is what happens when enzymes are affected by toxins and poisons and that sort of thing. And we'll remember, for the functioning of an enzyme, the enzyme has a very specific active site. It's specific for a particular substrate or substrates. And when those substrates bind to the enzyme, they form an enzyme substrate complex, affects the shape and functioning of the enzyme, and that change leads to a reaction. Well, in competitive inhibition, a toxin or a poison, something like this, will actually fit into part of the active site and block it. That binding is usually permanent. It effectively kills the activity of the enzyme, and it kills it because the substrate can no longer bind. So the function of the enzyme is lost. And I gave you a classic example, and that is of cyanide. The substance cyanide is a competitive inhibitor in the respiratory chain that we're going to learn about today. It binds to one of the enzymes in the respiratory chain and blocks it. And on a cellular level, the organism ceases to be able to respire. And that is lethal. So that is an example of competitive inhibition. It is not a way in which the cell normally uses to control the activity of enzymes, but the cell can control the activity of the enzymes. In some cases, they can be switched on, but in most cases, the control is to actually switch the enzyme off. And this is the mechanism whereby that is done. An enzyme can have, apart from its active site, as you see here, it can have another site at which binding can occur there. That is also very specific, but it's not an active site. It's not actually going to affect the substance that binds there. It's not going to promote a reaction there. Instead, when a substance binds there, it alters the shape of the enzyme, and the enzyme is no longer has an active site. It has an altered site there, which cannot bind the substrates any longer, so it can't promote a reaction. This is not a permanent binding. This is temporary, and this continually falls off. If the concentration of the substance is high enough in the cell, then this enzyme will remain shut off. If the concentration of the substance falls very low, then the enzyme will be able to be activated again. So this is a temporary binding here. This is called an allosteric site. This here is an allosteric enzyme. The whole system is called allosterism. And what is happening is we have a non-competitive inhibitor here binding to the allosteric site that we call an allosteric inhibitor. So this is a reversible situation. And it's extremely useful for the cell. because it gives the cell a fine control, not only of the enzyme's activity, but a control of the pathway in which that enzyme is involved. So this is non-competitive inhibition or allosteric inhibition. And here's how it works. We've got here a simple metabolic pathway. It has a substrate, begins with 2 intermediates, and then a final end product, a final product. Each step along the way is governed by an enzyme specific for that particular substance. So our very first enzyme here is specific for our starting substrate. There it is there, whatever it is. So there, under normal circumstances, it binds there and the enzyme promotes a particular reaction. But this is an allosteric enzyme. It can be, its activity can be stopped by binding an allosteric inhibitor there at the allosteric site. When it does, the shape of this will alter and it will no longer be able to bind. Under normal circumstances, when the enzyme is operating, it promotes the production of an intermediate. Then the intermediate is picked up by a second enzyme, and another intermediate is produced in the pathway. That is picked up by the last enzyme to produce the end product. So if I bind something there to the allosteric site, and I stop this enzyme from working, I'm going to stop this entire pathway because there's going to be no intermediate A, no intermediate B, and no end product. Okay? So all I've got to do to switch this off is bind an allosteric inhibitor and shut it down. What is the inhibitor? Where does it come from? Well, it's usually the end product. It's the end product of the metabolic pathway. And here it goes here. It binds to that allosteric site and it shuts this down. This is called feedback inhibition. This end product is feeding back to silence the pathway that produced it. Why? Why do that? Well, remember what I told you. This is a reversible binding. And how long this enzyme is bound, it depends upon the concentration of the allosteric inhibitor. So if this pathway produces lots of end product and the concentration is high, then the enzyme will be switched off. If the concentration of this end product falls, usually because the cell is busy using that end product for something else, pulling it off, pulling it off, pulling it off. As the concentration of end product falls low enough, then the enzyme will spend more time switched on and it will actually promote this pathway. And the pathway will continue to produce end product. When the end product concentration rises high enough, it will switch off. It gives the cell a very good way of fine-tuning the activity of a metabolic pathway. In a way, the cell is monitoring the concentration of the end product. And as the concentration of end product rises high enough, it switches off the pathway because producing more is now a waste of energy and materials. Okay? It only wants to produce that end product when it is really necessary, when the concentration is low enough that the pathway should be running to produce more. So this is feedback inhibition, and it is a very important way of controlling a metabolic pathway. The other thing I should tell you is it's usually the first enzyme in the pathway, which is the allosteric enzyme. These are not allosteric enzymes. And the reason for that is, well, just imagine if this one here was the allosteric enzyme, then in order to produce the allosteric enzyme, we'd have to run through a major part of the pathway and produce what are probably useless intermediates. It's a waste of energy and a waste of material to do that. You rather shut down the pathway right at the very beginning so that you don't waste energy producing stuff that you don't need. I would just add, for most metabolic pathways, these intermediates can actually be useful elsewhere. Very often they can be bled off to be used in another pathway. And that was one of the ways in which we produced those branched pathways. This is one of the ways in which these metabolic pathways link together. But for the purposes of our example here, we're just considering a single chain of events here. Please get this functioning clear in your head. So the one last thing I want to tell you about enzymes is I told you before that 99% of enzymes in the cell are actually protein, and that is true. But there is a very important exception, and that is this substance RNA, ribonucleic acid. And ribonucleic acid is a nucleic acid, obviously, closely associated with DNA and the functioning of DNA and all sorts of things, but RNA has many functions inside the cell, not all of which we will discuss. And some RNA molecules are able to act as enzymes, and often they are very, very important. For a start, they are involved in the processing of other RNA molecules and in their functioning in the cell. That is the one. For example, they may be involved in cutting and splicing messenger RNA, which we'll hear about in eukaryotes. The other place where we are definitely going to encounter them is as ribosomal components. Ribosomal RNA is part of the ribosome. It is structurally part of the ribosome. And it has enzymatic activity, which is essential in the production of proteins. We'll learn exactly what it does later on, but it is essential in production of proteins, in protein synthesis. The ribosomes produce protein from messenger RNA, and that activity is governed by the enzymatic activity of RNA in the ribosome. When RNA is functioning as an enzyme, we refer to it as a ribozyme. Ribosomal RNA enzyme. A ribozyme is RNA molecule which functions as an enzyme. Okay, so we're going to start talking now a lot about energy and energy transformation inside the cell. And I have mentioned some of these things before, but they bear repeating because sometimes it's a little bit difficult to conceive of the nature of energy inside the cell and why we need these different components that have to be present for us to successfully transform energy and use it inside the cell. The first thing to understand is energy doesn't just fly around the cell like a lightning bolt from one side to the other. Instead, we make use of energetic systems within the cell, and we will transform energy. Remember, energy can't be created, it can't be destroyed, but we can transform it. We can transform it from one form to another, and that is what the cell does. Ultimately, the energy which the cell uses resides in electrons which circulate around atoms. And we need to get a couple of chemical principles clear in our heads because we're going to hear them over and over again. When we're dealing with electrons, well, I can actually take electrons from a substance and I can take those electrons, put them onto another substance. When I do that, I'm actually doing two things. I'm removing electrons from a substance. That is called oxidation. That is an oxidation reaction, to remove an electron or electrons from a substance. When I donate those to a substance, the substance that gains them is reduced. That is a reduction reaction. And those two reactions always occur coupled. You never have electrons just flying around this free in a cell. You passage them. You passage them from one substance to another. Oxidize one substance, you must reduce another. So here, for example, we have a substance here which has already been reduced. It has an electron, which we're going to take off and we're going to donate. So this is called a half reaction. We're going to take the electron off and we oxidize that yellow substance. That electron is moved, doesn't just fly free, it is actually moved to another substance B, and that half reaction reduces B. So this is the reduced form of B. So that we're going to see over and over and over again. These paired reactions, oxidation, reduction, referred to as redox reactions. Oxidation reaction paired with a reduction reaction. The point of this is that very often what we'll do is we'll take an electron which has associated with it a tremendous amount of energy. And each time we pass it over like this, it will lose some energy. And we can harvest that energy, some of the energy that is lost, not all of it. None of these things are 100% efficient, but we can harvest some of that energy and we can use it to do something biologically useful. That is, you'll see how this works. Okay, so in these redox reactions, we have that clear. The electron donor is the substance which is oxidized. Oxidize the substance, it will donate its electrons to an acceptor. The electron acceptor is reduced. And in biological systems, what we need to realize is that if we are going to do this, we're going to need a supply of electrons. And they are going to have to be associated with substances which are biologically useful to us. The richest source of and easiest source of electrons in any system is to use a hydrogen atom. The hydrogen atom is the simplest of all of the atoms. It consists of one proton and then around it a single electron circulating. That electron can contain a tremendous amount of energy, and it actually can be energized, can have energy added to it by various means. That electron can actually also be stripped off quite easily, provided it has a destination to go to. It can be taken off. And that electron can then be passaged. It can be passaged through a series of reactions, and as it goes, it loses energy, and that energy can be harvested to be useful biologically. So the way in which we do that is we'll take a substance which is already reduced. Okay, it is very often, for example, we can use a fuel with lots of hydrogens associated with it, lipids, sugars, something like this, right? That'll have lots and lots of hydrogen available, but we need to take those hydrogens, we need to put them into a useful form, and that we call reducing power. We need to have them in a form which we can use to reduce other substances and harvest energy from them. So we need reducing power, and that reducing power is usually in the form of this substance, NADH. It is a coenzyme, NAD+. It is the NAD+ coenzyme, which we will reduce. And we reduce NAD+ with an entire hydrogen molecule, not just an atom, with a molecule. And a molecule of hydrogen is 2 protons, 2 electrons. It's H2, 2 protons, 2 electrons. So we've got a fuel, let's say. We're going to strip off through a series of reactions. We will strip off an entire atom of hydrogen, proton and electron, bind it to the NAD, and we will also strip off one more electron. And because this is NAD+, we end up with a neutral molecule, NADH. And NADH is carrying 2 electrons, which we can use. Very, very useful. In the process, we're going to generate a hydrogen ion, and a hydrogen ion is just the proton, H+. And you need to remember something about that we talked about before, about aqueous solutions. If I add lots of protons to a solution, H+, I'm going to make that solution acid. And that's what we're going to see happening. We're going to start accumulating hydrogen ions in the cell, and locally, at least in various places, especially at membrane, we can start to accumulate them and make that a little bit more acid. This carries some important implications that we'll discuss. So we take an organic molecule, we strip off its hydrogens, we oxidize it, and we reduce an electron carrier. We produce reducing power. And we can now take this and we can slot that reducing power into a series of reactions where we will harvest the energy of the electrons and do something useful. Okay. First thing we need, reducing power. So the second thing we need to remember is that energy, biologically useful energy, comes in the form of a phosphorylated nucleotide. A nucleotide that has lots of phosphate groups associated with it. The commonest, the most widely used in the cell is adenosine triphosphate, ATP. And if we look, here's ATP here. It's a nucleotide. Remember the structure of the nucleotides? They have a sugar. They have a nitrogenous base, in this case, adenine. And then they have phosphate groups associated. They can have up to 3 phosphate groups associated. And adenosine triphosphate is fully phosphorylated. And this here is actually the part that we're really interested in, is this last phosphate group with its bond here. And I'll tell you that the energy in that bond has been derived from electrons, from the energy of electrons, and we'll see how that is done. So there's a lot of energy in that bond. And if I break that bond, I can actually harvest that energy and I can use it to do something useful. Maybe I want to build a molecule. I want to make covalent bonds between monomers, let's say. I've got to put in energy. I'll harvest that energy from ATP, and I'll harvest it from here. I should add that under certain circumstances, we can also harvest here, but that's much more common that we see just this bond is used. And ATP is broken down by hydrolysis, as these molecules always are, and put in a water molecule. We break the molecule up and we'll harvest energy, right? And we'll be able to take that phosphate off and do something useful with it. Perform some activity, build something, perform some work, whatever. Biological systems almost invariably acquire their useful energy in the form of ATP. I say usually because there are occasions when you can use other nucleotides like guanosine triphosphate, but we probably won't see those. They're rare, and you usually see it being used in the form of ATP. What we're left with is adenosine diphosphate there. That's our raw starting product. And we're going to see in a series of very elaborate reactions, we can actually take a free phosphate and add it back in by putting in a lot of energy into this bond. We can recreate ATP. So put in the energy, we create ATP, temporarily store energy there, harvest that energy, and we can do something useful with the energy. So if I add a phosphate group, it's called phosphorylation. And there are various ways of phosphorylating ADP. If I take away a group, it's dephosphorylation. Okay, but phosphorylation, there are different ways of doing it. The one thing to note, however, is that ATP is not a long-term storage product for energy. It is a very, very temporary form of easily available, biologically useful energy. But we can't store our energy as ATP for very long at all. And if we want long-term storage, then the energy has to be put into stable covalent bonds. For example, the stable covalent bonds in glycogen, which is glucose units all joined together, or in lipids. Those are our very long-term storage products. But then when we want to access that energy and use it biologically, we can't just break a sugar molecule and grab the energy and use it. Instead, we have to put it through an elaborate series of processes where we will recreate ATP to use in our reactions. And that is essentially what we're going to be talking about today. So I need to first of all make clear there are various ways that we can take ADP and add a phosphate and form ATP. And this is called phosphorylation, as I've told you. There are three ways. The first is called substrate-level phosphorylation. This is an ordinary chemical reaction. We will simply take phosphate and add it chemically to the ADP and form ATP. The second is oxidative phosphorylation. This is an elaborate system. This is bacterial and our system of cellular respiration. And in cellular respiration, it is called oxidative phosphorylation. In actual fact, it's a useful name because it will remind us that I said to you, our electrons and the protons as well can't forever exist just free from one another. Eventually, they have to be joined together again. We have to form the hydrogen atom yet again at the end of all of our permutations. And in this system, oxidative phosphorylation, cellular respiration, as we'll see in a while, oxygen is actually the terminal electron acceptor. That electron that we've used all through those systems, these systems, to do phosphorylation, we're going to see that that electron eventually joins up with oxygen and its protons, and it forms H2O, water. That's one of the end products of this whole process. Oxidative phosphorylation we'll discuss in some detail. This system here, you won't recognize that name, photophosphorylation. Well, photo, light. Photophosphorylation is a method of producing ATP from light energy and using light energy to produce ATP. That is photosynthesis. This is photosynthesis. Light plus simple compounds, carbon dioxide and water. The light energy is used to actually produce, ultimately, to produce complex organic substances. But in the process of doing so, we form ATP containing the energy from the light, and some other substances as well that contain energy from the light. But ATP is one of them, and that would be photophosphorylation. Okay, let's have a look. This is just to explain more clearly the substrate level phosphorylation. And it's ATP made when we harvest phosphate from another substance, added to ADP by a simple straight chemical means, often using an enzyme intermediate, an enzyme intermediary rather. So we just take a phosphorylated fuel, perhaps, something like that, phosphorylated substance. We're going to grab its phosphate and add it to ADP and form ATP. This we're going to encounter very soon, because this is one of the reactions that takes place right at the beginning of our systems of oxidative phosphorylation. We have one pathway which represents substrate phosphorylation, and that is the process of glycolysis. So this is one of the reactions in glycolysis. The substance here, never mind everything else, just have a look here, phosphate, and then we, using an enzyme, we take ADP, we take that phosphate there, strip it off, add it to the ADP and form ATP, and we're left with this substance here, which now lacks that phosphate there. So this is a simple example, substrate phosphorylation. Oxidative phosphorylation is much more complex, and we'll discuss it in some detail. But here is an example. of how important that reducing power of NADH and FADH2 are. The NADH, FADH2, obviously, they are carrying hydrogen. And what we're really interested in is component parts of that hydrogen, the proton, and then these highly energetic electrons. And this is what oxidative phosphorylation does. Using a complex enzyme system embedded in the membrane, it runs those electrons from NADH and FADH2 through a whole series of oxidation-reduction reactions, a whole series of redox reactions. As the electrons pass through each of these, they lose energy, and that energy is harvested to produce ATP. So that energy is used to produce ATP. We'll see exactly how in a minute. At the end of this whole series of reactions, those electrons have to join up again with one another, and the way in which we do that is we bind them to oxygen. So we're going to see that's an entire hydrogen molecule. So that's 2 protons, 2 electrons will be run through the system at the same time. And at the very end, they are joined up to an oxygen atom to form H2O. So that's the final that's a byproduct of this entire system. And at the same time, we use up oxygen. That is why you breathe. That is why you breathe oxygen. It's to fund this process in your cells. Bacteria use exactly the same system. If they are aerobic bacteria, as I'll mention in a minute. Okay, so this is used in bacteria. It's used in everything else as well. Eukaryotes just as much as prokaryotes. So this is oxidative phosphorylation, a series of oxidation-reduction reactions in which the energy of the flow from the flow of electrons is actually harvested. This is photophosphorylation, and this is photosynthesis. And I'm going to do this in a little bit more detail later on, but not in much more detail. In photosynthesis, there are really, really specialized molecules called the pigments, which are chlorophyll. And there are actually 2 molecules of chlorophyll involved. There are some other pigments as well, but we won't worry about it. But chlorophyll is able to trap light, and some of its electrons are very, very loose, very easily excited by light. So the light energy here is converted into energy in the electron, and the electrons fly off the chlorophyll, and they are passed through an electron transport chain, much like we saw for oxidative phosphorylation, and ATP is created. But that's not actually the most important thing about photophosphorylation. This electron here, effectively the electrons are boosted again with more light on the second chlorophyll molecule, and this time they form reducing power. And this is a substance specific to plants, NADP, very similar to NADH rather. And it's in fact forms NADPH, that is reducing power for the plant. And the point of this is that these substances here, ATP and NADPH, are then used to bind, the energy in those is then used to bind carbon dioxide to water to form a sugar. So all of this energy here is originally light energy, and it's turned into a useful reducing power, chemical energy, and then is used to bind carbon dioxide and water. That's the process of phosphorylation. All right, so let's have a look at some specific pathways of energy production. And these are mostly metabolic pathways, so they are metabolic pathways which are varying complexity and structure as well. We're going to see a straight normal pathways, but then we'll also be introduced to a cyclic pathway. and a cyclic pathway, the end product of a cyclic pathway is the starting point as well. So you'll see it regenerates itself the whole time and then spits off products during the cycling. And all of these pathways that I'm going to talk about up to when we get to photophosphorylation, these are all pathways which are harvesting energy from organic compounds. So they are going. We're going to start off our discussion just realizing that we're going to use glucose as our starting product, our starting substrate, simply because it's the very easiest and it's a very common metabolic substrate to burn up to produce energy. So we're going to see how this substance is actually broken down. Now I can tell you, if I had a dish of glucose sitting on the table, I could actually light that glucose. I could burn it. And it would, if I burned it, what would happen? Well, it would burn, it would produce light as a flame, and it would produce energy. I would see that the energy coming off as light, but I would also feel energy coming off as heat. Where did that energy come from in burning glucose? Well, it's all locked into the bonds between the carbons, carbon to carbon, and between carbon to hydrogen. And it's those bonds, especially the carbon to hydrogen, that we are particularly interested in. And we're going to see exactly how we can access them. So the very first way is through a simple straight metabolic pathway, again determined by enzymes, each step of the way shepherded along, catalyzed by a specific enzyme, remembering that in these reactions, enzymes are unchanged by the reaction themselves. They're reusable and they work very fast and they can be reused over and over and over again. So we only need a little bit of enzyme to do a lot of reactions. And the very first set of reactions that we're going to talk about is glycolysis. And glycolysis is a process which does not use any oxygen. This is an anaerobic process. In some organisms, this is all that they do. Right? This is how they generate their ATP. It does not generate very much ATP, but in some organisms, like the anaerobic bacteria, those are bacteria which are able to operate without the presence of oxygen. Some bacteria are required. They are obligate anaerobes. They absolutely cannot have oxygen near them. Others, many bacteria, are facultative anaerobes. They can switch from one to the other. They can, and when they are anaerobic, this is the pathway that they are using. But all organisms use this as the starting point for the metabolism of substances like glucose. So we're going to start off with glucose. I do not expect you to remember the names of all of the intermediaries and everything. I expect you to be able to recognize the process and tell me what the products are and what the basically what we have to put into the system and what we get out of it at the end. Our starting substrate in this case is glucose. We can start with other substances. We can use fats or other sugars, etc. We may have to put them through other metabolic pathways to get them to a state where they can be put into this pathway. But we're going to use glucose. It's the simplest, that's the simplest thing to think about. So this is called, this first part here is called the energy investment step. And that is because glucose is an extremely stable molecule. It doesn't spontaneously break down very easily. And we need to actually kind of destabilize the molecule in a way. We're going to actually add energy to the molecule and destabilize it at the same time. And to do so, we're going to use ATP. We're going to use up 2 molecules, in fact, of ATP. And by doing that, we're going to add phosphates to the glucose. Well, how many? We got 1, 2, 3, 4, 5, 6 carbons. Glucose is C6H12O6. We've got 6 carbons. And we're going to add 2 phosphate groups to them. And here they are. Here, there's a phosphate, and there's a phosphate. And this forms a phosphorylated form of the sugar fructose. So the only thing that you need to know about this is that this is rather unstable. It is very easily broken up by enzymes. And we are going to break it into 2 parts. Don't worry about this. This is, in fact, when it initially breaks, it breaks into 2 different substances, and then this substance is converted to that substance. So don't worry about it. Just think that it is broken into 2 molecules of this glyceraldehyde 3-phosphate. Okay, sometimes just abbreviate G3P, glyceraldehyde 3-phosphate, G3P. 2 molecules, get that straight in your head. 2 molecules of G3P. I emphasize that because you see a pathway here, and you need to realize that this pathway is duplicated. It's duplicated because we actually make 2 molecules of G3P. So we've invested 2 ATPs, right? And we ended up with 2 molecules of G3P. Now we're going to put it through this pathway here, and we're actually going to start harvesting energy. We're going to harvest energy from this substance, glyceraldehyde 3-phosphate, in a series of reactions, which are going to end up producing pyruvic acid. That's going to be the end product of these reactions. So here, don't worry about the names of the intermediates, but have a look here and see what we actually make. What are the products of glycolysis? First of all, we're going to reduce NAD. We end up with 2 molecules of NADH, reducing power. Remember how important that was, how we absolutely have to have reducing power. We're putting it into energetic, we're taking energetic electrons, putting them into a carrier, which is going to be taken somewhere to do something useful with that energy. In addition, we're going to make 4 molecules of ATP. Why do I say 4 molecules? Why do I say 2 molecules here? Because we put 2 glyceraldehydes through here. Okay, look here and you'll see, don't get confused. This is the sum total. 2 molecules of NADH, 2 molecules of ATP, 2 molecules of ATP, 4 molecules altogether from the total reaction, and we invested 2. So our net gain is 2 molecules of ATP, 2 molecules of NADH there. Okay. And this molecule, 2 molecules of this at the end, pyruvic acid. Why 2? Because our fructose split into 2 molecules of glyceraldehyde 3-phosphate. Each of them went through their own pathway. This shows you the sum total there. We created 4 molecules of ATP, but we used 2. Our net is we gain 2 molecules of ATP, 2 molecules of NADH. For some organisms, this is enough. They can survive with this. They can survive with this as their sole source of ATP. They don't usually metabolize very fast. Usually the metabolism is much slower, but they can survive perfectly well. And many of them do. I just tell you, in evolutionary terms, we believe this was the first pathway that evolved. And for a very long time, this was the only pathway available to make ATP. There's one other thing I want you to notice, though, because I'll return to it at the end of this discussion, and that is to have a look here. Look at that molecule there, NAD. Remember what we said. This is critically important, that we have a source of that we can reduce to produce reducing power. We need NAD the whole time. Here we're using it up. Under normal circumstances, that's fine. We regenerate it. but there is a problem when we only use this as our only source of energy. We're only using up, we're not regenerating. So just keep that in mind for the moment. Okay, what do we use? We use up 2 molecules of ATP, glucose. We use up NAD, produce 2 molecules NADH. We produce 4 molecules of ATP by substrate phosphorylation. Each of these is a substrate phosphorylation reaction. And 2 molecules of pyruvic acid. That's for glycolysis. That's where it ends. I will just mention this is gone into in some depth in your text, but I won't do so. There are a couple of other pathways which microbes have. Just remember the names. Entner-Duderoff pathway, that is the one. It ends up producing slightly different products, ATP, NADH, and NADPH, which is unusual. And then there's another one called the pentose phosphate pathway, and it also ends up producing ATP and NADPH, not NADH. I'm not going to discuss the details. I'll just tell you that whether or not bacteria have pentose phosphate, Entner-Duderoff, or glycolysis only pathways is an important distinguishing character between them. And there are tests which we can perform in the laboratory to see, and we can use those to distinguish one bacterium from another. So I don't need you to know the details of those pathways. All right, so let's now talk about this elaborate system of cellular respiration. Respiration, we should think of as being a respiration. No, it's not. In metabolic terms, respiration is actually the use of oxygen in this kind of pathway to produce ATP. And this is a very efficient way of producing ATP. In our glycolysis, we only produce a very small amount of ATP, only 2 molecules of ATP. That's still useful. Some organisms can live with that, but it's not very much. Instead, by using this system of cellular respiration, of oxidative phosphorylation, we can actually produce a very big amount of ATP. And it's the way in which we're going to see it done is that we're going to use what's referred to as an electron transport chain. It's that series of redox reactions all linked to one another. And they are linked by the electrons which pass through them. As those electrons pass through, the electrons shed energy which is harvested. And it's harvested to form ATP. At the end of that pathway, we can't just have the electrons fly off by itself. Instead, we need to somehow reunite it with the protons from which it was stripped. And we do so in this system by using oxygen is the terminal electron acceptor. Now, there's one thing you need to know about oxygen. It is extremely electronegative. It is very, very, very attractive for electrons. It pulls electrons towards it, and it is one of the driving forces for this electron transport chain, is that at the end of it, we have this very, very, very strongly electron-negative atom sitting there, attracting electrons towards it. It actually pulls them through that chain. And finally, we bind the hydrogens to the oxygen, and we produce water as an end product. So there are several steps to this. First of all, here is our glucose in glycolysis. Here's what we produced. We produced 2 net ATP, and we produce NADH. We also produce 2 molecules of pyruvate. We're going to take each molecule of pyruvate, and we're going to put it through a little intermediate step. Very important step, though, because it produces NADH, more reducing power. And we then, by the way, as a byproduct, we're going to produce carbon dioxide from that reaction. There it goes. That's our first source of carbon dioxide. This is one of the reasons you breathe out carbon dioxide. You breathe in because you're using oxygen as a terminal electron acceptor. You breathe out carbon dioxide because you create it at various places in this process. We're going to push these through it's a metabolic pathway, which is a cycle. It's called the Krebs cycle, and they're named for the person who elucidated it. In the process of going through the Krebs cycle, we're going to generate more ATP on its own. Again, that's substrate level phosphorylation, more carbon dioxide voided as a waste product. But most importantly, we're going to produce masses of reducing power, masses of reducing power. And that reducing power, we're going to send off to this process called oxidative phosphorylation. That this reducing power, we're going to send to the membrane. And we're going to strip these electrons off all of this stuff. And this here, I'm going to strip off all of the electrons. We're going to do something very useful with the protons that are left. The electrons are going to send through that electron transport chain, and we're going to harvest all of the energy to produce, in the end, a whole lot of ATP. There's just one thing I want to tell you. You'll see various numbers for ATP. As it says 34 here, 38. Don't worry about it. Just get one number in your mind, and that is 32. That is the classic standard amount of ATP. It varies a bit depending on how you do your sums, and also on where in the pathway you slot in your electrons. There's various things. Get into your head, 32 molecules of ATP are produced as a result here of the final result of oxidative phosphorylation. Okay, so let's see. I just need to move something. Okay, remember the end product of the glycolysis was pyruvic acid. So here's the pyruvic acid here, a little 3-chain carbon. And what happens is that we're actually going to react this pyruvic acid with a coenzyme. It's called coenzyme A. And when we do that, we produce a molecule of carbon dioxide, but we produce this substance here. It's called acetyl coenzyme A. At the same time, we strip off hydrogen and we produce NADH. We produce some reducing power from this intermediate step here. Okay, and this is also this here is the source of carbon dioxide. Remember, we are duplicating this. We have 2 molecules of pyruvic acid to do this too. Okay, each of them will go through their own step. Now we enter what is called the citric acid or Krebs cycle. Citric acid cycle because citric acid is the very first product. We're going to react this 2-carbon compound here with a 4-carbon compound, oxaloacetic acid. Oxaloacetic acid and acetyl coenzyme A are the first substrates. They are reacted together enzymatically to produce citric acid. We then don't worry about the intermediates. Remember that oxaloacetic acid, citric acid, acetyl coenzyme A, and pyruvic acid. Remember, pyruvic acid, acetyl coenzyme A, joins to oxaloacetic to form citric. So that's why it's called the citric acid cycle. Now we're going to run it through a whole series of reactions, a whole lot of different reactions here. And what we're going to see, I just need to move something, sorry. What we're going to see is a whole series of reactions where we make reducing power especially importantly. We do make some ATP as well, which is important for the cell. But the most important thing about the Krebs cycle is its production of reducing power. So have a look here. This for each cycle. Here is NADH, we also produce some carbon dioxide. That's you breathe out. Here's another one, NADH, carbon dioxide. Here is a little part where we produce ATP. There's molecules ATP. Here we produce FADH2, that is reducing power. A little bit different to NADH, but functions in much the same way. Here is another source of NADH. How many have we got? 1, 2, 3, 4 in this whole diagram. 4 molecules of NADH and a molecule of FADH2 and a molecule of ATP. Double it, because we run this pyruvic acid through 2 of these cycles. Okay. This is what we need to watch. That's what we need to watch. Remember your sources of carbon dioxide, this intermediate step, and then the citric acid cycle. All right. Now we're going to follow these, the reducing power. Why are we following them? Why are we interested? Because they have hydrogen on them. We're going to take that hydrogen off. We're going to ionize the hydrogen. We're going to use the electrons, and we're also going to use the protons. We're going to do something very, very, very important with the protons. So here it is here. I just refer you back to the simple diagram. All that we do is this is all embedded in the membrane. Now in microbes, in bacteria rather, this occurs at the cell membrane, because that's the only plasma membrane. So this occurs at the cell membrane. Glycolysis takes place in the cytoplasm. But this electron transport chain is dependent on the membrane. These proteins and the enzymes that are embedded, that are responsible for this chain, they are embedded in the membrane. So this occurs at the membrane. And at the membrane, we're going to strip off. We are going to oxidize our electron carriers. And we're going to take those electrons and we're going to shunt them through this electron transport chain. Remember, it's attracted. They are attracted by this oxygen at the end. And at the same time, we're going to actually use energy to pump hydrogens. We're going to move hydrogens. And here, this is the way in which it's done. I think, yes, I have. Again, it looks very complicated, but I just want you to have the basic, simple understanding of what is in fact happening. Okay, so here, this is in a microbe, in a bacterium. This is the cytoplasm, and this is the outside of the cell. In a eukaryote, this takes place inside the mitochondrion. This takes place at the inner membrane of the mitochondrion, but the process is pretty much the same. So here's the cytoplasm. What have we got produced from all of our other reactions? We've produced masses of NADH. Here it comes here. We've also produced FADH2. They slot in slightly differently in the electron chain. Here's the membrane here. And embedded in the membrane, there are a whole lot of different proteins and associated substances, which are able to shuttle those electrons. And they shuttle the electrons from one to the other, from one substance, from the first substance to the next, to the next, to the next, in a series of oxidation-reduction reactions. So as the substance accepts the electrons, it is reduced. As it gives them up to the next substance, it is oxidized, so on and so on. They have some very special properties, though. And that is, some of them are actually able to pump protons. Now, remember what we're doing here. We're stripping off electrons, sending them through the system. We are left with protons. Protons begin to accumulate in the cytoplasm. And some of these substances here and here and here are able to pump protons across the membrane. they actively pump them, and they are pumping them using the energy of the electrons that are passing through. That's how the electron energy is harvested. It is turned into this kinetic energy, which actively pumps protons to this side of the membrane. Well, if I'm accumulating protons on this side of the membrane, I must have fewer protons on that side, right? Protons are positive. So I'm increasing the amount of positivity on this side. I'm increasing the positive charge on that side, and I'm decreasing it on this side here. I've created a differential, and that differential is separated by a membrane. That membrane is an insulator. It insulates against the passage of the protons. So we accumulate charge and we accumulate a chemical difference. There's a different chemistry. There are different ions on this side to what are on that side. The drive is to allow those to equalize, right, by diffusion. Similarly with the charge, if we accumulate positive charge, there's a less positive. This is more negative. This is more positive. Those tend to equalize one another, want to equalize one another. They can't. There's a membrane in between. What have I done if I do this? I've created potential energy. What do I call it if I have an insulator and I have masses of positive charge on one side and negative charge on the other side, and they can't pass between them across the insulator? What do I call that? It's a battery. That's how a battery works. A battery works by having an insulation layer. And on one side, we accumulate positive charge. On the other side, negative charge. And if we put a wire from one side to the other, the charge flows through the wire and it can do work with it. The battery has potential energy on either side of its insulators. Put a wire between them and we can harvest that potential energy and we can get it to do work for us. Light a bulb, turn a fan, whatever we want to do. The similar system here. We have an insulator. We're harvesting the energy of electrons and using it to pump the protons derived from the original hydrogen atoms. We accumulate charge and we also accumulate a chemical difference. Both of those are strong drives for these to try to move across here if they possibly can. So that sets the whole system up. But how do we make ATP from this? We've set up our battery. How are we going to use the battery? Well, we come here to this absolutely amazing enzyme. And this is called ATP synthase. ATP synthase spans the whole membrane, and it has a proton channel. It has a proton channel that runs through the middle of it. And when that channel is opened, the protons will flow very rapidly through the ATP synthase, and that causes the ATP synthase to manufacture ATP. That's what it does. It takes ADP and phosphates and it turns it into ATP. And this here is a chemiosmotic system. It's an osmotic system. It's hydrogens flowing through, and ultimately they'll form water. It's hydrogen ions, protons flowing through in response to the chemical and charge difference that we have allowed to build up on either side here. It's a very cunning system. We strip electrons off protons. We harvest those electrons and shunt them through these redox systems here. We harvest the energy and we use the energy to pump the protons, which were part of the hydrogen atom, across here. We build up charge and chemical difference, and then we allow that to equalize by having it flow through an enzyme which manufactures ATP. It's a wonderful system. These are just further elaborations on it for your especially for when you study. I don't expect you to remember all of the intermediates, but remember remember that there are those which not only harvest the electrons and pass them on, but when they do so, harvest the energy and use it to pump protons. So we have a series of proton pumps. And eventually we have a proton channel through the enzyme, which actually manufactures that actually manufactures the ATP. So now look where we are. We're at a stage now where we have the protons are flowed through here, and we have our electrons flying through this. Electrons can't be allowed to just be free. They can't do that. Instead, the electrons are strongly attracted to an oxygen atom. And at the same time, the protons are present. So what we have are protons, we have electrons, and we have an oxygen atom. Put those all together, and what we end up with is the molecule water. So that's the end product of this whole chain of events, are molecules of water. Oxygen is the terminal electron acceptor. It's actually attracting these electrons, and as soon as they get near, they're combined together with protons here, which are flowed through, and the hydrogen atoms are placed together again, proton, electron, two of those plus an oxygen atom, and we've got a molecule of water. So this is the budget for this whole process. This is the one I want you to memorize. Okay, I want you to know what products are at each stage of the process, and this is the one you can stick with. 32 molecules of ATP is our final total. And I think I will leave it there for today, and I'll complete this at our next lecture.